In this video, we are going to talk about experiments, introducing some common types of designs, and discussing some important challenges. First, let's start with a reminder. In observational studies, we record data without attempting to influence the response variables. In contrast, in an experimental design, different conditions are imposed on different sets of individuals, specifically to see how those conditions might affect the response variables. As a result, we are able to control some influential factors that could otherwise have become confounding variables. Because the conditions are imposed at random, the impact of those conditions is not associated with other factors. It's just random, which means that if we do see differences from one group with this condition and another group with another imposed condition, we are much more likely to be able to reach a conclusion of causation. It's important, though, to understand that our conclusions will be limited to individuals like the ones used in the study, placed in an environment like the one of the study. Our ability to control the environment is what can make experiments very simplistic or even unrealistic. At the core of an experiment is the ability to compare the effect of different imposed conditions on a response variable. And that means that an experiment must have more than one imposed condition. We may compare any number of treatments, a baseline or a fake treatment, also called a sham or placebo, or the absence of treatment, that is the most fundamental definition of the term control. Our ability to learn something interesting about those different imposed conditions rests on a lack of bias in the design. That means, first of all, that we must use several individuals for each of the conditions. Otherwise, the one individual would be confounded with the condition and we wouldn't be able to conclude anything interesting. And it also means that we must impose the conditions on sets of individuals chosen at random. It's because the conditions are imposed at random that they are not themselves associated with confounding variables. Another issue that we'll want to think about in designing an experiment is the bias, conscious or unconscious, of all the individuals involved in the experiment. Bias may come from the experimenters, or it may come from the subjects, or both. We can prevent bias by blinding to the details of the study. Only one of the two, subjects or experimenters, is blinded. We call that a single blind, and if both subjects and experimenters are blinded, we call that double blind. Obviously, blinding a study does not mean withholding informed consent. Informed consent is a very important ethical issue, and it should not be ignored. Blinding also allows us to study the effect of a fake treatment, the placebo effect. If the participants knew that they were getting the fake rather than the real treatments, then we wouldn't be able to tell whether a fake treatment has any kind of impact. Now, some comparisons can be a little bit tricky. Imagine, for instance, like this study, that you want to compare the effect of a medication taken in the form of a pill with an acupuncture treatment that requires placing needles in the skin. You can create a double dummy. Everybody took some pill and received some form of acupuncture. It was either the real pill with the fake acupuncture or the fake pill with the real acupuncture. Lack of realism can be a big issue with experimental studies, and it can take several forms. The individuals that you use for your experiment may not be quite representative of your intended target population, and the conditions might not be realistic enough to be applicable to real life. It's important to understand that conclusions based on sample data will only expand to individuals like the ones in your sample under conditions like the one of your experiment. A lot of medical studies, called clinical trials, are performed on male subjects, avoiding, if possible, the use of female subjects, perhaps out of concern over potential pregnancies. If you approve the medication to be prescribed for both men and women, then you have no idea whether your medication actually works for women. A lot of human behavioral studies will use made-up games to try to understand human behavior, but in real life, there are consequences that you cannot find in a game. So whatever you learn from those games at the lab might not actually truly reflect real-life circumstances and real-life choices. So we're going to describe a few common types of experimental designs. The first is the simplest and most common design. It's called the completely randomized design. And it gets its name from the fact that the individuals in the experiment are assigned to the different conditions completely at random. In the process, we end up with groups of individuals receiving different treatments that are independent random samples. That means that the individuals in those samples are completely unrelated. A variant of that is called the block design, where individuals are first split according to some feature before the random assignment. The purpose of this being to avoid imbalance in your random assignment that could possibly occur by chance. In both of those designs, the comparisons we make will happen at the group level. In contrast, we may design an experiment where we want to be able to make comparisons at the individual level. This kind of experimental design is often called a match pairs experiment, although you will see other kinds of naming conventions such as repeated measures or crossover experiment. So we're still talking about an experimental design in which conditions are imposed. But in this case, the conditions are imposed on pairs or sets of related individuals. So if you have actual pairs of individuals, like twins or siblings, then within each pair, you may assign at random one individual in the pair to receive one treatment and the other individual to receive the other treatment. Alternatively, if you're using the same individual for all treatments, then each subject would be tested under each different condition at a different point in time. That means that you would end up with repeated measurements for each subject. The advantage of a design with matched pairs or repeated measurements is that allowing the comparisons to be made at the individual level can be useful to detect subtle effects of your imposed conditions. Here are two examples. Use what you've learned so far to match each study with its corresponding design. First of all, notice that both studies are experimental studies. We know that because in both of those studies, the participants did not decide what they were going to do when. The choices were made for them on purpose as part of the experimental design. In the first study, two different conditions were imposed: eating version A of a new chili ball versus eating version B of the new chili ball. We have repeated measurements here because each participant had to eat and rate both chili ball versions. The comparison in ratings can therefore be done at the individual level. The order of the food presentation was randomized to avoid the order becoming a confounding variable. We all know that food tastes much better when we're hungry than when we're full. So it's important to make sure that the same version isn't always presented first. In the study, two different treatments were imposed. We know that the design was a completely randomized design because our pool of 140 patients was simply assigned to these two conditions completely at random. We end up with two independent random samples. Therefore, the comparison can only be done at the group level between those two samples. So we've seen some types of experimental designs, such as, most importantly, the completely randomized and the matched pair experimental design. They are very common types of designs, but keep in mind that they are not the only kind. Experimental designs can get very complicated, but the more complex a design is, the more complex the resulting data analysis is going to be.